So hello everyone. As was said, I'm mostly a biomolecular scientific archaeologist. However, don't worry, I'm not going to go completely sciencey and crazy with this presentation, so it will be understandable. So I'm going to be presenting today the results of some of the biomolecular analysis we've been doing at the site of Lochhead Quarry, which is the medieval long-kissed cemetery in Angus. And this is part of a collaboration with a whole bunch of people, as you can see from so all of the authors here, that is not everyone involved in the project, that's just the authors. My general PhD project is a collaboration between the labs we have at the University of Aberdeen, so with Kate Britton and Lena Skurdlin-Flink, with museums and special collections there, and then also with the Francis Crick Institute. And as mentioned, it's pre- and proto-historic Scotland. What this has ended up meaning is that I've mostly ended up studying Pictish stuff, but I've looked at Neolithic stuff as well that's not in my thesis because of time. And my main focus is on isotopic and genetic analysis, focusing mostly on site-specific analysis and the individuals there, ensuring these don't get lost in kind of the big border narratives you often see publicized. So unfortunately today, I'm not going to be presenting some fun population genetics. I'm going to be presenting stuff that's a lot more localized, but hopefully still interesting. So what do we mean by life histories? So I actually focus on quite a specific area of that, which is the scientific analysis of human skeletal remains and all the different things from them that we can tell about that individual, building up a bit of a biography of them. So these are the various ways we can look at the skeleton when we excavate it using scientific methods. So we've got things like our oxygen and strontium, which we do on teeth. The M2 there is that means your second molar. So that looks at things like residence. We've got isotopes that we do from the bone collagen that we look at things like diet. Obviously radiocarbon dating. Endogenous aDNA, what I mean by that is stuff from the person themselves. So not things like pathogens, but we can look at that. And things like osteology. So today, oh, wait, where's it gone? I'm missing a slide here, but there I'm going to be mostly looking at biological relatives, genetic sex, residence and diet in the sort of last decade or so of life, this residence, CalBP, and then things like stress and trauma and age at death. We'll go through these as we go along, so don't worry about remembering all of them. And I'm doing this for a site in okay, there are quite a few slides missing. This is fun. So this is a site called Lochhead Quarry, which was excavated in 2004 by AOC. So this is sort of the landscape around where it was. This is some pictures of the quarry. This is not actually from the excavation. And this is just some of the things I've been able to find online. Currently, it's still a working quarry. So they originally found one kist that was out at the bottom of the quarry face. This was excavated, believed to be prehistoric. And then a couple of months later, more kists emerged, and it turned out to be a wider cemetery. So this is the rough view of the site with the plan. And so this is from Dunbar and Maldonado in 2012. So this is the main kind of publication I've got most of my information from. So this site is actually just outside Forfar, near a place called Octoforfar Farm. And there are a total of 20 long kists that have been excavated from there. So as you can see, kist 20 up here was the first one to emerge. And then 1, 2, 4, and 8 were probably the ones that were next spotted. The crosses here indicate those that were very much coming out of the quarry face. So 1, 2, 4, and 8, I think, were all actually lifted as a block and excavated out of situ, but still in situ because of the kind of dangers faced by the archaeologists doing this. And as you can see, they were all this kind of rectangular stone-lined kist. There were 20 in total, only 18 of these housed skeletal remains that were present. However, that's still really good for Scotland. So this is part of the reason why we've chosen this site for analysis, because we've got a lot of lovely individuals that we can actually look at, as opposed to other sites where you get body stains or empty graves. So about the site organisation, there's no intercutting, as you can see, which suggests there may have been grave markers or some deliberate awareness of the location of previous graves. And they're all in this sort of east-west generalised alignment. There is the potential deliberate clustering of graves, which I'll come back to later, here, and then with 20 out to the side. It's possible that this cemetery was originally bigger, but this is kind of the whole excavated area and they didn't see any more. The two kists that didn't have individuals in them are believed to be non-adults potentially based on size. Take that with a grain of salt because that's really hard to infer because they originally thought that kist 20 might have been a crouched up individual, stuff like that. And the main interpretation of the site previously was that this was a family burial ground. So these are the radiocarbon dates. So 10 of these were from the original publication, which are just recalibrated. And then we also got KISS 9. We got a date from the Finlay Harris-Dick Pictish Prize for Research, which I'll come on to why we chose KISS 9 later, which shows a distribution mostly from about 400 to about 700. So about 300, 250 years from the 5th to 7th centuries. So my research is looking at what connects the burial community and what can we learn about the people who were buried here, trying to answer the elusive question of who were the Picts. Spoilers, I don't have the answer to this. So we're looking at various things and I'll go through them in this presentation. So things like lifestyle, so that's sort of things like, for example, stress that you experience throughout life, any trauma, any illness. And we look at that through osteology. So this was done originally at AOC and then Jenna Dittmar had a look at that at Aberdeen. Genetic kinship, i.e. were these individuals biologically related, which we do aDNA for. Lifetime mobility, so is where the individuals were buried different from the place where they were likely living during childhood. And for that, we need about 3 to 8 years of age. For that, we're using strontium and oxygen. Diet, what were they eating? Carbon, nitrogen, sulfur. And where may have they been living in later life? And that's sulfur. And I'll come back and go through that as we go through. So we analysed all of the individuals we could for all of these different things where we could, but it depends on what you have left. So for DNA, we were able to sample 17 of the 18 individuals for genetics. For strontium and oxygen, we were able to look at 14 because you need to have the certain tooth. For these ones at the bottom, we were able to look at all individuals because all you need is a bit of bone for that. So generally, the preservation at the site was really poor for general standards of skeletal remains, although pretty good for Scotland. So this just shows a picture of the sort of conditions we're working in, and that's a pretty good one, I think, there. So it was really hard to make any osteological observations on the skeletons. From what we were able to tell, there were males and females, and there was a bias towards younger adults, so sort of before the ages of 45. But it's really hard to tell that because it's based on things like teeth, which aren't always there. What we can also say is that there was poor dental health, stress, and an active lifestyle. So there was also quite a lot of things like osteoarthritis. And in particular, they had some really nice, clear instances of this, which is what we call a linear enamel hyperplasia. So you see these lines on the teeth. So no. These lines here, you can see them on some of the others. And that's what happens when, as your teeth are growing, if you have periods of stress. For example, this could be malnutrition. It could be having illness. So my dad has some of this from getting scarlet fever as a kid. You get these lines. And this could potentially be used to indicate that these individuals were of lower status in Pictland. Take that with a grain of salt, but that is another bit of evidence alongside the fact that this is not a monumental cemetery. So DNA, a boring, beautiful graph here. So the first thing that we found was that there are far more females than males. It's actually a ratio of about 2 to 1. So we have 12 females genetically, with one a bit spurious, 5 males, and then 1 which we don't know because we weren't able to do DNA. And this pretty much matches up with the osteology, but a lot of them were unknown. This individual here, it's in brackets, the female, because there wasn't enough DNA to be really confident. So of what we have for DNA, as the beautiful science is, it takes a very long time. So we only have 12 of the individuals that we sent off fully sequenced. So these ones at the bottom that are in brackets, we are waiting for more information on. This one we're not going to get because there's not enough there, but the other 4 we should. So what all of this means, which looks very scary and confusing, is essentially we have a bunch of different mitochondrial haplogroups, and this is what you get from your maternal line. So on 23andMe, everyone in this room would have one of these, and then not everyone's going to have the Y. Pretty much all of the ones that we're confident on are different. There are some repetition, but the variation in this suggests that we've not got a nice set of matrilineage at this site, which if there was any chance that we really thought this was a strong thing in Pictland, this is kind of showing it's not, at least at this level. And the male haplogroups are all incredibly consistent with what we'd expect at this time period in Scotland. The real surprise, which we were absolutely not expecting, is that there are no genetic relatives amongst the 12 that we've got the full sequencing for. And this is up to 3rd degree or closer, which essentially means up to great-grandparents, cousins, great aunts and uncles, things like that. And that's amongst all 12 individuals. So none of those are really closely related at all. It's possible that amongst the other 5 or others that we haven't got, there are relatives, but even so, this is completely unexpected, considering the previous interpretation was that this was a family burial ground. And this analysis was done by Marina Silva at the Crick. Oh, okay. Right. A lot of these slides are missing, so I can't show you some nice maps. We did strontium and oxygen analysis for the 14 individuals. And of those, around 8 of them were consistent with the area around the site. And what that means is essentially they're eating food and drinking water that was procured in the nearby region. Doesn't guarantee that they're from there, but that's what we'd perhaps call local. But in this case, the actual extent of the region of Scotland we're talking about is quite large. And there's no associated settlement with the site. So it's very difficult to kind of say where. And another 2 of those individuals, so in the area we're talking about, there's kind of an area of what you might call lowlands, and it goes up into the Cairngorms. 2 of them then have strontium and oxygen, which is more consistent with living up there in the Cairngorms, or at least getting food from there. So they're sort of nearby the site in that you could probably get there within about a day, but still quite distinct. So this is not just one village or settlement community. We're talking about at least during childhood. So people are coming here during life. We then have a further 4 individuals that are absolutely not local. And we can tell that definitely because they have oxygen, which is from a much warmer environment. And this is probably further towards the west coast. So it's variable between those 4 individuals. About 3 of those, including KISS9, and we got this data back before we did the date, which is why we chose to date it, are most consistent with the west coast of Scotland. So it could be somewhere in the region of Dalriata, or it could be further up into sort of Atlantic Pictland. We also have consistency with things like Shetland and Orkney, so there's the possibility of coming from there. And one of those individuals has quite high concentrations of strontium, which is a bit unusual, and that can be associated with Maca soils. So that also is further evidence that it'll be from the west coast or possibly the Hebrides, but not quite the Hebrides because of the oxygen. The final individual, KISS20, which, as you'll remember, is the one that is much further out. This individual is from the areas that it could have been, this individual could have grown up are really small. And so there's a small area in Aberdeenshire and then a bit further to the south of Scotland. There are also a large large bits of Ireland, in specifically Northern Ireland, where this person could have come from, which is again consistent with what we can see. But that's a bit more limited. So what you have in terms of the mobility is people from a lot of different places coming in, which is very consistent with the DNA. In terms of the diet, so this is a plot. So all these little crosses here are individuals from Lochhead, and then the blue is individuals from other sites across Pictland, and then this is just the mean. And this is one of the reasons we're originally drawn to the site, is this is what we got from the radiocarbon dates for carbon and nitrogen, which can be a little bit dodgy. This is very low, so we wanted to retest them. And as you can see, they fall fairly similar to most other contemporary Pictish individuals. So they're eating a diet probably of things like sheep and goat. You have got some variation within this though. For example, these two individuals up here, and then a bit further down here. But the problem with this type of analysis is that you need to compare it to things like animal bones, because this will vary in different places across Scotland. So therefore, what seems low at one site might actually be quite high compared to the fauna at that site. We don't have that for Lochhead because it's just an isolated burial ground. We don't have a settlement. But they're not super out of the ordinary, but they are a bit lower than other sites. Yeah, so sulfur is thoroughly consistent with them living in Lochhead Quarry region in later life. This does not guarantee it, it just means they might have. So going back to these questions here, what might have connected this burial community? Was it genetic kinship? Absolutely not. Most of them, or pretty much all of them, are not related. So even though they might be kin, they're certainly not genetic relatives, which is what. Lifetime mobility, they're not all from the same place. Quite a few of them might have been in the local region, but various places within that. So they're not growing up together, they're moving to come to this site. They're eating similar-ish things, which could bring them together at that point in later life. And again, because they could well have been living nearby, but we don't know, it's very possible that this is a community that is coming together later on in their lives and then being buried in a nearby region. And this is sort of backed up in the lifestyle. We've got similar levels of things like stress, which could suggest a sort of shared social grouping or class maybe. But really what actually has happened with this biomolecular analysis is we had quite a nice, neat, defined site, and it's become incredibly confusing and weird. And this mirrors, and a lot of it mirrors what we're seeing widely across early medieval sites in Scotland, which is these high levels of mobility, this similar diet, and then also, for example, having more females is kind of commonly observed across sites. It also fits with the wider observations that we're seeing of high levels of mobility in these, obviously we've heard today, like these big high status sites, probably people moving around the landscape more. We're seeing this at multiple levels. And it is also very possible, and a lot of this work has been emphasized by Adrian Maldonado, that actually these sites are a method of bringing people together and reinforcing these ideas of community, and emphasizing new groups that are being created. So actually the possibility that, you know, these people have very different life histories is actually not surprising, because this is a mode of bringing people together rather than necessarily them having that shared, absolutely same living in one village for their entire life. So yeah, still work ongoing, very open to ideas and contributions and ideas, things that people think about all this. But I specifically want to thank AOC, and specifically Alexandra Johnston at AOC has been brilliant at helping provide me with information, and also all the folk at museums and special collections have been very enthusiastic and wonderful and helpful. So great, thank you to them. And that's some of my reference. Oh, there they are. There's the missing slides. So this is the maps, and that's the area. So this is what we mean by the cairns. So yeah, beautiful maps. Sulfur, yay. These are the thanks. There we go. Thanks, everyone. Thanks for